Hi, it's Miss Barnhart, and let's continue our discussion in genetics by looking at some extensions in Mendelian genetics. Remember that Mendel was the Australian monk who was studying pea plants. During his investigations, he discovered the law of dominance, the law of segregation, and the law of independent assortment. However, very few traits are expressed in simple dominant recessive patterns. First, let's take a look at incomplete dominance. Incomplete dominance is when the heterozygous individual exhibits a third phenotype that's in between the two homozygous phenotypes. An example is the color of flowers in snapdragons. In this case, there's a red allele and a white allele. Flowers that have two red alleles are red. Flowers that have two white alleles are white. But if you have one red and one white, red doesn't dominate, it's pink. Incomplete dominance alters the phenotype ratio we see because now the genotype ratio is equal to the phenotype ratio. Second, let's take a look at codominance. This is similar to incomplete dominance, but a little bit different because in this case, the heterozygote condition expresses both of the homozygous conditions simultaneously. Think of it this way. In incomplete dominance, a red allele and a white allele together were expressed as pink. In codominance, a red and white allele would be expressed as red and white striped. Both alleles would show up. An example of this are the A, B, and AB blood types in humans. If you have an A allele, then you make A glycoproteins on the surface of your red blood cells. If you have a B allele, you'll produce B glycoproteins on the surface. But if you receive an A and a B allele, you produce both A and B glycoproteins. The body makes anti-blood type antibodies for any carbohydrate that is not on the surface of its own red blood cells because these carbohydrates act as antigens. These antibodies cause human cells that have that particular carbohydrate on them to agglutinate or clump. People with type A blood will have the A antigen on the surface of their red cells and anti-B antibodies in their plasma. If type B blood is introduced, agglutination will occur. Anti-A antibodies are present in a person with type B blood, but neither antibody is present in a person with AB blood, and both are produced in a person like me with O type blood. This combination of antibodies makes it possible for us to type someone's blood as seen in the picture here. Can you see that receiving the wrong blood type can be deadly? Another way that Mendel's results are varied is when there are more than two alleles for a trait. This can occur in two ways. First, if the gene locus has more than two possible alleles for the trait, and second, if there's more than one gene that controls the inheritance of that trait. The first case is called multiple alleles. More than one gene is known as polygenic inheritance. The best known human example of multiple alleles is the ABO blood type. In this case, there are three blood type alleles. This results in 6 possible genotypes and 4 phenotypes. A and B are codominant to each other, and they dominate over little I. This chart shows how the blood groups are formed. Polygenic inheritance, on the other hand, results when you have more than one gene contributing to the expression of a particular trait. We generally see these traits exhibited as a continuum of phenotypes. Two well-known examples are human skin color and human height. We know of at least three genes at different chromosomal locations that are responsible for skin color. This leads to a wider variation of phenotypes along a bell curve distribution. There's a wide variation between extreme phenotypes, with most individuals having intermediate phenotypes. In skin color, the dominant genes are additive, with each dominant gene adding one unit of color to the genotype. The more dominant genes, the more the phenotype appearance gets darker. Now let's take a look at how genes can interact with each other. In these complex patterns of inheritance, the interactions between the genes in the organism can be quite variable. Pleiotropy refers to one gene having multiple phenotypic effects on the physiology of an organism. One example of this is sickle cell anemia. One locus has wide-ranging effects on the physiology of the entire organism. Can you see the sickled shape of the red blood cell here? This was caused by inheriting two abnormal alleles for hemoglobin, which cross-linked instead of forming the normal globular shape. The normal red blood cell shown here contains normal hemoglobin, which can carry oxygen to body cells. However, a cell containing abnormal hemoglobin has this characteristic sickled shape. It can't carry oxygen and it clogs up small blood vessels. Untreated, it can lead to pain, swelling, and possibly death of tissues or a stroke. This shows that all these effects are directly due to the inheritance of the sickle cell allele. The deformed red blood cell shape is directly due to the inheritance of two alleles for the abnormal hemoglobin, and this leads to the multitude of effects. Now let's take a look at epistasis. In epistasis, two or more genes control the expression of a single trait. One example of this is the inheritance of coat color in Labrador retrievers. This involves the interaction of two different genes. One, we'll call it big E, determines whether or not pigment is produced. If a dog inherits at least one dominant allele, then this allows the gene for pigment color to be expressed. In this gene, the capital B allele is dominant to the little b allele. Dogs with one or more big E's inheriting one or two big B alleles are black, while dogs with one or more big E alleles inheriting two little b alleles are brown. If a dog inherits two little e's, he'll be white, regardless of which combination of the big B alleles he inherits. The phenotypic ratio deviates from the 9:3:3:1 expected as a result. That's epistasis. And I hope that's helpful.